Good evening, dear students. So, uh, yesterday's class, we have started with the new topic that is biomolecules, right? So, we have discussed a few things, right? So, we have discussed what are biomolecules, right? Then we had started with carbohydrates. We have discussed the what are carbohydrates. Then we have discussed classification of carbohydrates. Then we have discussed about glucose, right? The structure of glucose. So different reaction to confirm the presence of different groups. So these all things we have discussed in our previous class, right? So any doubts? Yes, any doubts? So these all things we have discussed. Okay. So today we are going to see the remaining part. So let us start now. Yes. Okay. Fine. So in the last class, we have discussed about the structural illustration, right? So in that, there is something called a D and L form. Okay. So we are going to discuss something called as D and L configuration. Okay, D and L configuration. So D and L configuration. So you can see when we, okay, I'll write one example before that. So CHO, C, OH, H, and CH2OH. Okay, so this is a molecule, same molecule, but we have written it on the different sides. Okay, you can see the different sides here. So here what you can see that, okay, so this is the molecule glyceraldehyde. Okay, so which is the molecule? It is glyceraldehyde. What is the name of the molecule, students? It is glyceraldehyde. So both these are glyceraldehyde only. Okay. But what you can see, what is the difference? What you can make out? That in this first, okay, here the OH group is on the right hand side and here the OH group is on the left hand side. Right. So this is also glyceraldehyde. Now, when we see some of the compounds, okay, you can see they have written something called as D like this, right? And they write L. Some letters are written, right? D, L. So what it means, okay? So these letters, that is D and L, okay, they are written before the name of the compound. Okay, what it indicates? So D and L, capital D and L. Okay, so these are the letters which are written in front of the compounds. Okay, name of the compounds. What this indicates? This indicates the relative configuration. Okay, what it indicates? It indicates the relative configuration of a particular stereoisomer. Okay, of a particular stereoisomer. So, this is the meaning of the letter. Okay, so we are understanding or we are seeing the relative configuration. Relative configuration means what? The relative arrangement, okay? The arrangement of different atoms. So we can see the arrangement of different atoms and this different arrangement can be understood or it can be represented using this capital letters D and L. Okay. Now, for assigning this D and L letters, okay, we are going to take a standard. So this is a standard which we have taken, that is glyceraldehyde. Okay, so we have taken glyceraldehyde as a standard compound. Now here you can see it consists of one asymmetric center. Now what do you mean by asymmetric carbon or asymmetric center, students? Anybody? Yes, we have discussed these all things, okay, in haloalkanes and haloarenes. What do you mean by asymmetric center? or a chiral center. Yes. So this carbon, okay, the carbon is attached to four different atoms or four different groups. Okay, so this carbon is said to be a asymmetric carbon or chiral carbon. Okay, so this will exist, okay, in this two forms. Now here you can see that when the OH is on the right hand side, okay, when the OH is on the right hand side, then it is called as diglyceraldehyde. Okay, it is represented as diglyceraldehyde. If OH is on the left hand side, then it is represented as what? It is represented as L-glyceraldehyde. Okay, so remember this. So when OH is on the right hand side, it is represented as D. When OH is on the left hand side, it is represented as L. Okay, so this is your DNL configuration. Suppose, okay, suppose if I write the structure of glucose now. Okay, now let us write the structure of glucose. Okay. Now here what you can see, how many carbons are there, students? So one, two, three, four, five, six. Six carbons. Okay. So here you can see about this, the OH is on the right hand side. The OH is on the right hand side. So this is D plus glucose. Okay. So this is D plus glucose. Okay. So this is the configuration. Understood? So this is our D and L configuration. So when the OH is on the left hand side, it is called as what? L. We we give the letter as L, and when OH is on the right hand side, we give it as D. Okay, so this is the meaning of the D and L configuration. Okay, now let us see next part. So next part we are going to see the cyclic structure of glucose. Okay, we are going to understand the cyclic structure of glucose. So how the cyclic structure of glucose is given. Okay, we know the glucose, how it is represented. Yes, students, how glucose is represented in yesterday's class. We have discussed the structural illustration of glucose. So carbon, okay, which is having a aldehyde group, then carbon having, okay, a hydroxy group. So this is the arrangement. And lastly, a primary alcoholic OH group. Right. So here, so this is a representation. So 1, 2, 3, 4, 5, and 6. So 6 carbons are there, right? So these are 6 carbons. Let me write it 1, 2, 3 only. Okay. So let me write it as 1, 2, 3, 4, 5, and 6. Okay, fine. So when we are writing the cyclic structures, okay, when we are writing the cyclic structure, so the cyclic structure, so what will happen? It will form a hemiacetal structure. So this is called as a hemiacetal structure. Okay. So here what will happen? Cyclic hemiacetal structure of glucose is formed. Okay. And how it is formed? It is formed between the OH group of the fifth. Okay, so fifth carbon, so the OH group of the fifth carbon, and it adds to your carbonyl group of the first carbon. Okay, and it forms a six-membered ring. Okay, how it forms? Let us see. Let us see. So how it forms a six-membered ring, students. Let me show you. So here, so the bond is formed between the fifth OH of the fifth, okay, and to the first carbon, between the first and the fifth. So the bond, it is like this. Okay. So it will form a hemiacetal structure. So this is our hemiacetal structure. So let us write. So CHOH, then CHOH, CH, CH2OH, and okay, like this. So this is what? This is your hemiacetal structure of glucose. Now here you can see OH is on the right hand side. So it is alpha D plus glucose. Okay. Now here if the OH attacks from this side. Okay. If the OH is on the this side, then it forms either it can be on right hand side or on the left hand side. So this is the another structure. So hemiacetal structures are very important. They will ask you for exams. Okay, so please practice these structures. Very important. So again, the bond is formed between the fifth carbon, okay, and the first carbon. So this is your hemiacetal structure if OH is on the other side. Okay. So here again, so 1, 2, 3, 4, 5, right? And okay, so this is a six-membered ring, right? Six-membered ring. So here you can see that this is a, so this name is what? Beta D glucose. Okay, so this is a beta D glucose. So it can exist, okay? It can exist in two cyclic hemiacetal form. So this is the hemiacetal structures of glucose. So here the OH group is on the left hand side. So it is beta D glucose. Okay. So if it is on the right hand side, so it is alpha D glucose. So this is the cyclic structure of glucose. Now here, this two cyclic structures, okay, everything is same. Yes, students, everything is same in both the structures. This and this structure, if you compare, all the number of carbon atom, number of hydrogen atoms, oxygen atom, the bond formation, everything is same. What is the only difference? The only difference is at the C1 carbon, right? Only difference is at the C1 carbon. Here, in the beta D glucose, the OH group is present on the left hand side, and in the alpha D glucose, the OH group is on the present on the right hand side. Okay. So here the difference. Okay. So if there is a cyclic configuration, if there is a change in configuration at only C1 carbon, okay, only at C1 carbon, then such isomers are called as anomers. Okay. Anomers are those isomers, okay, which will show difference in the configuration at only C1 carbon. Okay, at only C1 carbon, right? So these are the two isomers. So what is the difference? What is the change in configuration? The change in configuration is only at the C1 carbon. So such isomers are called as what? Anomers. Okay, is it clear up to here, students? Is it clear? So we have discussed about the cyclic structure of glucose. Okay, we have seen the hemiacetal structures, how it differs. Then we have studied something called as anomers. Okay, what is anomers? So when there is a difference at only C configuration, change in configuration at only C1 carbon, okay, then such isomers are called as anomers. Okay, so these anomers can be best represented in their Hayward structures. Okay, one more important thing, Hayward structures. So Hayward structure can be asked for 2 marks. So they can ask you to write the alpha D glucopyranose structure or beta D glucopyranose structure of glucose. Okay, so let me write the Hayward structure. I hope you are writing down the structures. Okay, so it will be helpful for you. So when we are writing down the Hayward structure, so after seeing the structures, okay, so this is the HOH CH2H. Okay, so this is your alpha D glucopyranose. Okay, and your okay. Now here, let me show you how I have written this structure. So let me write the alpha D structure. So COH, okay, HCOH, HOCH, then HCOH, CH, and CH2OH. Now here, so this is also our alpha D glucose. Okay, alpha D glucose structure. Now here, what you can see, so this is which is our C1 carbon. So C1, C2, C3, C4, and C5 carbon. Okay, and C6 carbon. Right. Now here, which is our C1 carbon. So here, okay, C1 carbon is this one. C1 carbon. Okay, so here you can see this is on the right hand side. So this is on the right hand side. Then this is C2 carbon again on the right hand side. At the C3 carbon, this is on the left hand side. So here. Then C4 carbon again on the right hand side. Right. And again CH2OH is on the right hand side. So again here above and here it is like this. Okay. So like this is our alpha D structure. Okay. So this is our Hayward structure of alpha D glucose. So this is our Hayward structure of alpha D glucose. Okay. Got it? So this is the Hayward structure. What is it? Hayward structure. So now I'll write the Hayward structure for your beta D glucose. Now beta D glucose, what is the difference? Here it will be OH and here it will be H. So seeing that, let us write the structure. So first draw a six-membered ring. Okay, so your OH will be above, H will be here, then your H, OH, then will be OH, H, then will be H, OH, and here it will be CH2OH and H. So C1 carbon, C2, C3, C4, C5. Okay, and C6. Got it? So like this. Is it clear? Is it clear? So this is what? This is our beta D glucopyranose. Pyranose. Okay. So this is our Hayward structure. So they may ask you to write the Hayward structure for alpha D glucopyranose or beta D glucopyranose. So you need to remember the hemiacetal structure. So if you know hemiacetal structure, so depending upon the hemiacetal structure, you can write the Hayward structure. Is it clear up to your? Any doubts? Any doubts up to your? Okay, fine. Others? So we have completed the uh, glucose. Next, we are going to study the fructose. Okay, next carbohydrate that we are going to study is fructose. Okay, so fructose, what is the formula? C6H12O6. Okay, so this is a ketohexose. So this is example of what? Ketohexose. Why ketohexose? Because it consists of a ketone group, right? It consists of a ketone group. So how it is obtained? It is obtained when we do acid hydrolysis, okay, of sucrose. So if we do acid hydrolysis of sucrose, what we are going to get? We are going to get 1 unit of glucose and 1 unit of what? 1 unit of your C6H12O6, C6H12O6. So 1 unit of glucose and 1 unit of fructose. Right. So this is when you get it from sucrose. Okay, you are going to get when you do acid hydrolysis of sucrose. Okay. So this is the glucose and fructose, what you are going to do. So when it undergoes the acid hydrolysis, so this is the preparation. So from this you can extract your fructose. Fine. Okay. So this is a, so let me write the structure of your fructose. So the structure of fructose is CH2OH, C double bond O. Please note down the structures. Structures are very important, okay, and you need to practice all the structures. So H, OH, CHOH, and CH2OH. So this is a structure. So 1, 2, 3, 4, 5, and 6. Okay. So this is our D minus fructose. Okay. So this is our fructose structure of fructose. Now from this structure of fructose, I can write the hemiacetals. Okay. I can write the hemiacetal structures. So again, the bond is between the C5. Okay, it is between the C5 and C2. Okay, it is between the C5 and C2. So how it is formed? So it will form a four-membered ring. Okay, sorry, it will form a five-membered ring. Right, it will form a five-membered ring. So let us see. So if the OH is on the right hand side, so what you are going to get? So this will be, so after when it attacks here, so it forms OH, okay, COH, and here will be CH2OH. So this CH2OH will come this side, so CH2OH, okay. Now the C, here will be your HOH, so HOH, then CHOH, then HC, and here will be your bond, and here will be your CH2OH. So this is your alpha D fructose. So this is your what? Alpha D fructose. Now, if the attack takes place on the other side, so OH will come here. So it will be OH, C, CH2OH, then CHOH, CHOH, CH, and here is your, and this is your CH2OH. So this is what? This is your alpha, sorry, this is your beta. So this is your beta D fructose. Okay, so this is your beta D fructose. Understood? So your, what is the only difference? The only difference is at the C2 carbon. Okay, the only difference is at the C2 carbon. Okay, so alpha D fructose and this is beta D fructose. So this will exist in, so your fructose, it will exist in two forms. Okay, it will exist in the cyclic forms. And how it is obtained? It is obtained by addition of this OH group of the fifth carbon to the keto group. So if the attack is taking from this side, you will get alpha D glucose. If the attack is taking from the left hand side, then you will get the beta D glucose. So depending upon the attack, from which side it will take place. So these two cyclic forms are possible. Okay, so remember this, both the forms of alpha D and beta D fructose. So depending on this, let us write the Havoc structure now. So let me write the Havoc structure. Now this is a five-membered ring. So five-membered ring will be like this. Okay, so this is our five-membered ring. So this is our five-membered ring. So now again, I'm writing this structure, first one structure. Okay. Now your CH2OH is above, so it will be CH2OH. OH is right side, so this will come here. Then will be your OHH. Then will be your HOH. Then will be your CH2OH and H. So this is your first carbon, C1, C2, C3, C4, and C5. Okay. So this is your alpha D fructopyranose, furanose. Okay. Alpha D fructofuranose. Okay. So this is your this structure, Havoc structure. Now let us write the Havoc structure for beta D fructopyranose. Now depending on this, so your OH will be above. So this will come above and here will be your CH2OH. Then will be your HOH. Then will be your HOH. Here will be your CH2OH and H. Okay, so this is your what? This is your beta D fructofuranose. Okay, beta D fructofuranose. So these are the Hayward structure. So remember that cyclic structure, that is hemiacetal structure, and the Hayward structure. Okay, so these both are very, very important. Okay, so these are very, very important. Is it clear up to here? Is it clear up to here? So this completes our monosaccharides. So we have seen two important monosaccharides, which is that we have seen about glucose and we have seen about fructose. Okay, so we have completed the monosaccharides. Monosaccharides. Okay, now we are going to see about disaccharides. Okay, next we are going to see about disaccharides. Now, what are these disaccharides, students? What do you mean by disaccharides? So okay, what are disaccharides? So when the carbohydrates undergo hydrolysis, they will yield two units of, okay, they will yield two units. Okay, so here what will happen? Disaccharides. So these disaccharides, how they are formed? They are formed by the joining. Okay, if you take two monosaccharides and you link them. Okay, if you join two monosaccharide and if you link them, then you get a disaccharide. Now, the first example of a disaccharide which is there for your syllabus is maltose. Okay, it is what? It is maltose. So let me write. So what all important things are there? That much only I am going to cover. Okay. Now here, so maltose. So what is maltose? Maltose is also called as malt sugar. What it is called as maltose. I hope you are noting down these things. Okay, it is maltose is also called as malt sugar. What is the formula? It is C12H22O11. Okay, C12H22O11. Now, what does maltose consist of? Maltose consists of two, uh, um, uh, maltose consists of two alpha D, alpha D Maltose, what does it consist of? It consists of two units of alpha D glucose. Okay, it consists of two units of alpha D glucose. Okay, now how the linkage is present? So the disaccharide, it consists of one special type of linkage, and this linkage is called as the glycosidic linkage. Okay, it is called as glycosidic linkage. This glycosides linkage, it links between the two units. So between these two units, the link which is present, it is called as glycosidic linkage. Now, how the units are joined? Okay, let me draw. Let me draw. Please see the structure. Draw the structure as well as, okay. So glucose unit, you know the structure of glucose. Alpha D glucose units are present. So alpha means your OH will be down. Okay, so your HOH, then OH, H, HOH, and CH2, OH, and H. Then another unit, okay. So again, alpha D glucose, so HOH, then OH, H, H, OH, and CH2OH and H. So these are the two, two, three, four, five, six. Okay. So one, two, three, four, five, six. So see, these are the six carbon atoms. Now, how to link these two carbons? So these two groups, they are linked, okay, by a link which is called as the glycosidic linkage. So here both the OH are there, right? So what will happen? The linkage is present between which, here you can see the linkage is present between, so maltose, it is made up of two alpha D glucose unit. Okay, and the link is present between the C1 of one glucose unit. So this is one glucose unit, the C1 of one glucose unit, and the C4 of, okay, C4 of another alpha D glucose unit. So this is, you have to remember about maltose. Okay, this you have to remember about maltose. So maltose, what it is called as malt sugar, how it is made up of? It is made up of two alpha D glucose unit. And this is a structure of maltose. Okay, I hope you have noted down. Yes. Did you note it down? Okay, fine. So let us go further. So next disaccharide which is there for you is the sucrose. So sucrose is also the disaccharide. It is also called as cane sugar. Okay, cane sugar. The formula is C12H22O11. Okay, so what does sucrose consist of? Sucrose consists of alpha D glucose. Yes, sucrose consists of what? Sucrose consists of alpha D glucose and beta D fructose. Okay, alpha D glucose and beta D fructose. So these are the units present in the sucrose. So you know the reaction. Okay, so when you do acid hydrolysis of sucrose, right? When you do acid hydrolysis of sucrose, you get what? You get 1 unit of glucose and 1 unit of fructose. So this is your alpha D glucose and this is your beta D glucose, fructose. Okay, remember that. Fine. So let us see the structure now. So one is the glucose unit. Okay, you know the glucose unit, how to represent the glucose unit. Alpha means again OH will be down. So HOH, then here HOH, then OHH, then HOH and CH2OH and H. So this is the structure. This is the structure. HOH and this is CH2OH. Okay. Now this is one alpha D glucose unit. Now another is what? Beta D fructose. So how is beta D? Beta D means it is Furano structure. So this is a Furano structure. So beta means OH will come above and this will be CH2OH. This is OHH. This is HOH and this is CH2OH and H. Okay, now how the link is present? Okay, so when sucrose is formed, the bond is present between the C1, okay, of alpha D glucose. So this is the alpha D glucose. So this is the alpha D glucose. So the C1 of this and the C2. So C2 of the what? Beta fructose. So beta D fructose. So the link is present between what? This two. So OH and this OH, what you will get? You will get O like this. So this is the structure. Okay, so this is the structure. Okay, so what is sucrose structure? So sucrose structure, it is formed. Okay, and what type of link is this? This is a glycosidic linkage. Okay, so this is what? It is a glycosidic linkage. So this linkage is present between C1 and C2. C1 of alpha D glucose and C2 of beta D fructose. Okay, so this is and this is also called as sucrose is called as what? Non-reducing sugar. Okay, it is called as non-reducing sugar. Why it is called as non-reducing sugar? Yes, Why it is called as non-reducing sugar? Because it do not reduce, it cannot reduce tolerance reagent and Fehling solution, because there is no free aldehydic. Okay, there is no free aldehydic or no free aldehydic or ketonic group. So because of this, because there is no free aldehydic or ketonic group, sucrose acts as a non-reducing sugar, and it will not it will not reduce the Fehling solution and tolerance reagent. Is it clear? Is it clear, students? So this was the second disaccharide. Okay, this is the second disaccharide. Is it clear? Did you understand about sucrose? So what is sucrose? Sucrose is cane sugar. How sucrose is made up of? Sucrose is made up of one unit of alpha D glucose, another unit of beta D fructose. So this is a structure. Okay. So what type of linkage is present? It is a glycosidic linkage. So it is between the C1 of alpha D glucose and C2 of beta D fructose. And why sucrose is called as non-reducing sugar? Because it does not have free aldehydic or ketonic group. Okay, and it do not reduce tolerance and tolerance reagent and Fehling solution. Okay, so this is about sucrose. Let us go for the third one. It is lactose. So the third disaccharide unit is lactose. So lactose is also called as milk sugar. Okay, lactose is also called as milk sugar. The formula is C12H22O11. Okay, C12H22O11. So lactose, it is made up of what? It is made up of beta D galactose. Okay, so it is made up of beta D galactose and beta D glucose. Okay, it is made up of what? It is made up of beta D galactose and beta D glucose. Okay. So let us see the structure. So please write down the structures. Structures are very important. So this is beta D galactose. HOH, OHH, OHH. Okay. And beta D glucose, OH on the top, H below. Then again HOH. This is OHH. And this is HOH. And this is CH2OHH. Now the bond. So how to link this two? So the bond which is present, the linkage which is present, again it is a glycosidic linkage. Okay, and to join these two units, it is joined by a glycosidic linkage, and the bond is between the C1 of galactose. Okay, C1 of galactose and C4 of glucose. Okay, and C4 of glucose. So this is a C1. Yes, C1. So this is a C1 and this is your C4. So the bond is between this. Now, how it will form? Just see. So here is the OH, here is the OH. So it will form a, so this is the bond formation. Okay, so this is a bond formation. So this is the fourth one. Okay, so this is the glycosidic linkage which is present between the C1 of galactose and C4 of glucose. Yes, note down the structure. Note down the structure of lactose. So this is lactose. Lactose is also called as milk sugar. So what it is made up of? It is made up of beta D galactose and beta D glucose. Okay. So what all things you have to remember? Now we have studied three disaccharides. Okay, we have studied three disaccharides. The first one was what? Maltose. The first one was maltose. The second one is sucrose. Second one is sucrose, and the third one is lactose. Right. Now, maltose, it is made up of what? It is made up of two alpha D glucose unit. Okay, two alpha D glucose unit. Then the linkage is present between what? So maltose, what it consists of? Maltose consists of two alpha D glucose units. Okay. And the linkage is present between C1 and C4, which is the linkage glycosidic linkage. The second one is sucrose. Second one is sucrose. In sucrose, what it consists of? One is alpha D glucose and another is beta D fructose. Okay, beta D fructose. The linkage is present in between what? The linkage is present between C1 and C2. C1 of your glucose and C2 of your fructose. Okay, and the third one is lactose. Lactose consists of what? One beta D galactose. Okay, one is beta D galactose and another is beta D glucose. The linkage is present between C1 and C4 and this is a glycosidic linkage. So remember this, okay? Very important. You need to know the which are the monomer units, which are the monosaccharide units and what is the linkage present in that. Is it clear? Is it clear up to here? So we have completed monosaccharides, then we have completed monosaccharide and we have completed disaccharide. Next, we are going to move for polysaccharide. Okay. Okay. Next, we are going to study about polysaccharide. Now, polysaccharide means what, students? What do you mean by polysaccharide? Polysaccharide means it produces large number of monosaccharide units, right? So, when polysaccharides, when they undergo hydrolysis, when carbohydrates, when they undergo hydrolysis, they produce large number of monosaccharide units. And these units, they are joined together by glycosidic linkages, okay? They are joined together by what type of linkages? It is joined together by glycosidic linkages. So which are the different polysaccharide? The first one is starch. Okay. The first one is starch. Now, so starch is the polysaccharide unit. Okay. So what is this starch? Where it is present? It is present in plants. Okay. It is used or it is preserved or it is used as the storage, okay, storage polysaccharide, which is present in the plants, okay, and where it is for, particularly, it is present particularly in the roots, okay, in the tubers and some vegetables, okay, and in some vegetables. And what does the starch consist of? The starch consists of alpha glucose units. Okay, it consists of alpha glucose units, and the two components which are present in the starch is amylose and peptine. Remember this, very important. Please remember this. The polysaccharide component, okay, which is present in the starch, it is amylose and amylopectin. Okay, amylose and amylopectin. Fine. Now, what is this amylose? Amylose is water soluble. Okay, this is a water soluble component. Okay, and it mainly consists of about 15 to 20% of starch. So the most of the starch is just present in this amylose, which is water soluble. Okay, and this is long and it is a straight chain. Okay, it is long with a straight chain and what is happening? The alpha D glucose, they are joined in the what do you say, in the ring form, okay, in the straight chain form, okay, they are joined in a straight chain form. So like this, around 200 to 1000 alpha D glucose units, they are held, okay, and the type of linkages present between this glucose unit is the glycosides linkages. Okay, so what you can see that, so amylose, it is a water soluble. Okay, it is a water soluble component and it consists of 50 to 20% of starch. Okay, so how it is, what is the structure of amylose? It consists of, so it is a long chain like structure. Okay, it consists of long straight chain and it consists of about 200 to 1000 alpha D glucose unit. Okay, it consists of 200 to 1000 alpha D glucose units and these are held by glycosidic linkages. So this was about the starch. That is about amylose. Now, what is about amylopeptine? Okay, amylopeptine, it is a insoluble. Okay, it is insoluble in water. It is not soluble. It is insoluble in water and it consists of 80 to 85% of starch. Okay, it consists of 80 to 85% of starch. Okay, and as in amylose, it was a straight chain. Okay, it had a straight chain, long straight chain. Your amylopeptine is branched. Okay, your amylopeptine is branched. Okay, and here there is a branching. Here you can see C1, C4 linkage. Here the this is between C1 and C6. Okay, it is between the C1 and C6. So branching is present. So C1 and C4, again the branching is present between C1 and C6 like this. So amylopectin is branched compound and the structure of amylose is a straight long chain form. Okay. So this is about your starch. Next is cellulose. Okay. Next we are going to study is about cellulose. So what is cellulose? So cellulose also it occurs in plants. Okay. It occurs in plants. Okay. And most probably in the cell wall. Okay, the constituent is in the cell wall. So this is also a straight chain polysaccharide. So this is also a straight chain polysaccharide. Okay, and it composed of only beta D glucose unit. So how the in starch, what it consists of? It consists of alpha D glucose unit, right? It only consists of alpha D glucose units. And here, in case of cellulose, it consists of beta D glucose units, and they are joined by C1 and C4 glycosidic linkages. Okay, it is the the bond is formed between C1 and C4, and it is a glycosidic linkages. Okay, so this is about your cellulose. Okay, this is about your cellulose. So this much thing you have to remember. Next is glycogen. The third polymer, polysaccharide, which is there for you is the glycogen. So glycogen, where it is present in the animals. Okay, it is used as a storage. Okay, it is used as a storage. Okay, to store in the animal body, it is called as the carbohydrates which are present. It is stored in the animal body in the form of glycogen. Okay, so whenever the body requires any glucose, glycogen, okay, so the glycogen, what it does, it breaks down this glucose, okay, by the enzyme, and it will take up the energy. So this is it is also called as the animal starch. Okay. Why? Because it is the structure of this glycogen is similar to amylopectin. Okay. But glycogen is again highly branched. Amylopectin is what? It is branched. Okay. It is branched. It is having branch form. So your glycogen is highly branched. So you can see more branching in your glycogen. Okay. So where this glycogen is present, it is mainly present in the liver, okay, liver or in the muscles and in the brain. So, so this is so the high concentration, it is present in these particular organs. Okay. Is it clear? So these are the three polysaccharides which are there for your syllabus. Fine. Okay. So this was about your carbohydrates. Okay. So this was about your carbohydrates. So in carbohydrates, we have discussed briefly, okay, completely about monosaccharides. So in monosaccharide also, we have discussed two, that is glucose and fructose. Then we have discussed about disaccharides. Then disaccharides, we have discussed about maltose, then your lactose and sucrose. Then we have studied about polysaccharides. In polysaccharide, we have discussed about starch, cellulose, and glycogen. So this completes our carbohydrates. Next, we are going to discuss about proteins. Okay, next we are going to discuss about what? Proteins. Now, what are proteins, students? Yes, what are proteins? Can anybody tell me what are proteins? What are proteins? Anybody knows? What are proteins? What are proteins? Proteins are essential for our body, right? So proteins are what? They are the essential for human body. Yes, proteins are essential for the human body. So how they are essential for human body? Okay, so amino acids. So amino acids are the building blocks for proteins, right? Amino acids are the building blocks for proteins. Now, what are these proteins? Proteins consist of amino acid, and these amino acids are nothing but they are the nitrogenous compound, right? These are the nitrogenous compound, and these proteins, they are required. Why? They are required for the growth, as well as the for maintenance of the living organism. So proteins are required by the living organism for their growth, as well as for maintaining the life. Okay, so all the proteins, they are made up of the smallest unit, which is amino. Okay, that is a amino acid. So all proteins, they are made up of what? The building block of protein are the amino acids. So let us start with proteins. So in proteins, we are mainly discussing about the amino acids. So what are amino acids? Okay, amino acids are what? So amino acids, they are represented by R, CH, NH2 and COOH, right? So what you can see here, so this is a representation of your alpha amino acid. So here you can see, okay, so here you can see the amino acid structure. Here you can see, right? So here, so this is a one, so in the amino acid, what you can see, there is one amino group, one carboxylic group, and one side chain. Okay, so depending upon, so what it consists of one amino, one carboxylic acid group in the same molecule. So depending upon the position of NH2 group, okay, with respect to your carboxylic acid, okay, they can be classified as alpha, beta, gamma, and so on. Okay, now here you can see there is only one carbon, so it is designated as alpha. If there was one more carbon, then it would come as beta. Okay, so depending upon the position of this amino as well as the carboxylic acid group, okay, so the designation is given here. So this is the alpha amino acid. So what are amino acid? As I have discussed, so amino acid, they are the organic compound. Okay, in the same compound you are having two functional group, that is a amino group as well as the carboxylic acid group. Okay, now there are mainly 20 amino acids. So okay, so there are 20 amino acids. So the 20 amino acids, so which are the 20 amino acids? Okay, so the 20 amino acids are glycine, okay, and alanine. So please, in the textbook, okay, you can find out the structures of these 20 amino acids and go through the structures of all the amino acid. Okay, so please go through the structures of all these 20 amino acids. So let me tell you which is the amino acids. So glycine, okay, glycine, alanine, then you have valine, okay, then you have leucine, isoleucine, okay, and so these all are under a category called as neutral. Neutral means what? In a neutral amino acid, you have one amino group and one carboxylic acid group. So it is called as a neutral amino acid. So these are categorized as neutral amino acid. Next, we have arginine. Okay, we have arginine. We have lysine. Okay, these are termed as basic amino acid. Why they are termed as basic amino acid, student? Because we have extra one amino acid. Okay, extra one amino acid. So because of that, they are termed as what? They are termed as your this. So extra amino, your basic group is more there. So they are termed under basic. Okay. Next, you have glutamic acid. Glutamic acid. You have aspartic acid. Okay. You have glutamine. Okay. And you have aspargine and threonine. Okay. And threonine. So these are in acidic group. Okay. They are having in the acidic group. So these are the 12. Then remaining serine. Okay. Then cysteine. So serine, cysteine, then methionine. Okay, then you have phenylalanine, tyrosine, then you have tryptophan, you have histidine, and you have proline. So these are all 20 amino acids. Okay, so here they have categorized into different groups. Neutral. Okay, so in neutral, so these are coming in neutral. Acidic means one extra carboxylic group. So this comes under acidic. Then you have basic. Basic means one extra amino group. So these are the comes under basic. Then S containing means what? Sulfur containing amino acids are cysteine and methionine. Then alcoholic. Alcoholic means it consists of one OH group. So these comes under alcoholic amino acids. Then aromatic means what? The such amino acid, they consist of cyclic structures. Okay, so these are the aromatic amino acids. Then you have heterocyclic. Heterocyclic means what? In such amino acid, you have a nitrogen atom, okay, present in the ring structure. Okay, so these are the amino acids which comes under heterocyclic. So these are the classification of different amino acids. So please go through all the 20 amino acids and please study the structures. Please go through the structures of these amino acid. Okay. So, as I have discussed, okay, so the amino acids, they are classified on the basis of the nature of amino acid. Okay, so first one, they are classified as acidic, basic, or neutral. So, based on what? Based on the nature. Based on nature of amino acid. Okay, based on nature of amino acid, they are classified as what? Neutral, acidic, and basic. Now, what do you mean by neutral? Neutral means they have equal number of, okay, equal number of carboxylic acid and NH2 group. Okay, so which are the example? The example can be glycine, aniline, valine, leucine, isoleucine. So any one example you can give. So there can be question like this: what is neutral amino acid? So that time you have to explain with one example. Then what is the acidic amino acid? In acidic amino acid, okay, you have more number of carboxylic group. You have more number of carboxylic groups. Then they are called as acidic. Okay. Then which are the example? Aspartic acid, aspargine, glutamic acid, and glutamine. Next is basic amino acid. Basic, it consists of more number of amino groups. Okay, more amino groups are present. Okay, so which is that? Arginine, lysine, etc. Fine. So these are the first classification based on the number of nature of amino acid. Next. So next categorized is based on the biological importance. Okay. Next is based on the biological importance. So based on the biological importance, how they are termed as in based on the second classification of amino acids, second classification based on biological importance. Okay. So they are termed as the first one is essential amino acids. Okay. What very important, mark this as important. What the question can be, what are essential amino acid? Give example. Or what are non-essential amino acid? Give example. Okay. So the first one is essential amino acids. So what are essential amino acids? So the amino acids, okay, amino acids which cannot be synthesized by our body. Okay, which cannot be synthesized. Okay, which cannot be synthesized in the body. And this should be obtained through diet. Okay, this should be taken through diet. So you have to take it through diet. So such amino acids, they are called as essential amino acids. Okay. Essential amino acids are those acids which cannot be prepared or it cannot be synthesized in our body. We have to obtain this through the diet. Okay. And so these are the essential amino acids. Second one is non-essential amino acid. Non-essential amino acid means what? Yes, non-essential amino acid means this can be, okay, this can be synthesized in our body. Okay, this can be synthesized by our body. So which are the example? The examples are glycine, okay, then aspartic acid, then serine, okay, cysteine. So these are the examples of non-essential amino acids. So please make a note of these. What is essential amino acids and what are non-essential amino acids? What are essential amino acids? So these amino acids cannot be synthesized by our body, and it should be taken or it should be obtained through the diet. Okay. And what are non-essential amino acids? These are the amino acids which can be synthesized by our body. And what are the example? Glycine, aspartic acid, serine, cysteine, etc. Okay, is it clear up to here? Yes. Is it clear? Any doubts up to here? So we have discussed about the amino acids also. Okay, so the anything you have any doubts? Okay. So next one, this is isoelectronic point. Okay, next is isoelectric point. So please mark this as important. What is isoelectric point? What is isoelectric point? It is the pH of amino acid solution at which the amino acid, okay, it will neither move at cathode, okay, neither it will move to cathode nor it will move towards the anode. So it will be at one point. Okay, so the pH will remain as it is. It will not change. Either it will not move to cathode nor it will be moving to anode. So this is isoelectric point. So what is isoelectric point? It is a pH of amino acid solution, okay, where the amino acid do not move either towards the cathode or towards the anode. So if I give an example, so let me give an example of glycine. So glycine, okay, has a isoelectronic point of pH 6.7. So at this pH, okay, the glycine will neither move to cathode nor it will move to anode. Okay, so it will be at one particular pH. So at this pH, it will be at its isoelectronic point. Okay, then next term which is important is Zwitter ion. Next term which is important is what? Zwitter ion. Okay. Now what is Zwitter ion? Now RCH2 NH2 COOH. Okay. So now here what you can see, there is a hydrogen. Okay. What you can see here, there is a hydrogen. So this hydrogen from the carboxylic acid, it can, okay, it can be removed and it can form NH3 plus, right? So this is in its covalent form. This is in its covalent form and this is called as its Zwitter form. Okay, it is its Zwitter form or it is forming a Zwitter ion form. Okay, so what you can see here, your when you make the solution of this amino acid, okay, when you make the solution of the amino acid, your carboxylic group, okay, your carboxylic group, what it is doing, it is donating one proton. What it is doing, it is donating one proton to the amino group. What it is doing, it is donating one proton to what? It is donating one proton to the amino group. Okay, and it forms a dipolar ion. And what it forms? It forms a dipolar ion. Okay, and this state is called as a Zwitter ion form. It is called as what? Zwitter ion form. Okay, so what is done when the amino acid is added in the what aqueous form? Okay, so the carboxylic acid, what it is doing? It is donating the proton. Okay, it is donating the proton, and what it is doing? The amino acid will take the proton and it will form a dipolar ion. Okay, it gives a dipolar ion, and this is known as what? Zwitter ion. Okay, understood what is Zwitter ion and what is isoelectric point? So these are two important points, okay, which has to be remembered. So is it clear? Okay, fine. So this much is enough for today, I think. Okay, so we have lot more. So if I continue that, again, it will be more of confusion. So let us stop today till here. Okay. Remaining things I'll be taking in tomorrow's class. Okay. So what all things we have discussed today? Okay. What all things we have discussed today? We have started today. Yesterday's class, we have completed glucose structure. So today we had completed fructose. Okay. The monosaccharide one. Yesterday we have taken one example, okay, of monosaccharide glucose, and today we have taken fructose. Then we have started with disaccharides. So disaccharides we have studied three, three sugars. After that we have studied polysaccharides. So in polysaccharide we have discussed about starch, cellulose, and glycogen. Okay, then we have studied in starch about amylose and peptine, amylopeptine also, right? Before that we have also discussed about DNL configuration, right? And the cyclic structures of glucose, right? All the then Hayward structure of glucose, fructose, maltose. So these all things we have discussed. So remember all these things, okay? Don't forget. So please practice these structures. Then we have started with proteins. What are proteins? What are amino acids? Then we have seen the 20 naturally occurring amino acids. Then we have discussed about the classification of amino acids. Okay, the first classification was based on the nature of amino acid, acidic, basic, and neutral. The second was based on the biological classification, biological importance, that was essential and non-essential amino acids. Right. Then we have discussed about isoelectric point and Zwitter ion. Okay. Then tomorrow we will be discussing about peptides, polypeptides. Okay. Then we are going to study about proteins and the types of proteins. Okay. So these all things let us study in tomorrow's class. Fine. Okay. So we will be, so after proteins, we are going to discuss about enzymes. Okay. Hormones and vitamins. Okay. So then nucleic acids. So these are the things which are left. So that we'll be discussing in tomorrow's class. Fine. Okay. So I shall end the session here. Okay. So the remaining things we'll be discussing in tomorrow's class. Thank you.